16S ribosomal RNA is a component of the 30S small subunit of prokaryotic ribosomes. The genes coding for it are referred to as 16S rDNA and are used in reconstructing phylogenies, due to the slow rates of evolution of this region of the gene. Woese and George E. were two of the people who pioneered the use of 16S rRNA in phylogenies. Multiple sequences of 16S rRNA can exist within a single bacterium. Functions: it has several functions. Like the large ribosomal RNA, it has a structural role, acting as a scaffold defining the positions of the ribosomal proteins. The 3' end contains the anti-shined algano sequence, which binds upstream to the August start codon on the mRNA. The 3' end of 16S RNA binds to the proteins S1 and S21 known to be involved in initiation of protein synthesis. RNA-protein cross-linking by A.P. Zernilowski al. interacts with 23S, aiding in the binding of the two ribosomal subunits, stabilizes correct codon-anticodon pairing in the A site, via a hydrogen bond formation between the N1 atom of adenine residues 1492 and 1493 and the 2OH group of the mRNA backbone structure. Universal primers: the 16S rRNA gene is used for phylogenetic studies as it is highly conserved between different species of bacteria and rDNA. Woese pioneered this use of 16S rRNA. In addition to these, mitochondrial and chloroplastic rRNA are also amplified. The most common primer pair was devised by Weisberg al. and is currently referred to as 27F and 1492R. However, for some applications shorter amplicons may be necessary, for example for 454 sequencing with titanium chemistry the primer pair 27F534 are covering V1 to V3. Often 8F is used rather than 27F. The two primers are almost identical, but 27F has an M instead of a CAGAGTTTGATCMTGGCTCAG compared with 8F, PCR applications. In addition to highly conserved primer binding sites, 16S rRNA gene sequences contain hypervariable regions that can provide species-specific signature sequences useful for bacterial identification. As a result, 16S rRNA gene sequencing has become prevalent in medical microbiology as a rapid and cheap alternative to phenotypic methods of bacterial identification. Although it was originally used to identify bacteria, 16S sequencing was subsequently found to be capable of reclassifying bacteria into completely new species, or even genera. It has also been used to describe new species that have never been successfully cultured. 16S ribosomal databases: the 16S rRNA gene is used as the standard for classification and identification of microbes, because it is present in most microbes and shows proper changes. Type strains of 16S rRNA gene sequences for most bacteria and rDNA are available on public databases such as NCBI. However, the quality of the sequences found on these databases are often not validated. Therefore, secondary databases which collect only 16S rRNA sequences are widely used. The most frequently used databases are listed below. 1. EZTaxon. http://estixon-e.etsbyallow.net/. The EZTaxon database is an extension of the original EZTaxon database. It contains comprehensive 16S rRNA gene sequences of taxa with valid names as well as sequences of uncultured taxa. EZTaxon contains complete hierarchical taxonomic structure for the domain of bacteria and rDNA. 2. Ribosomal Database Project http://rdpcmeMSU/edu/ The ribosomal database problem is a curated database that offers ribosome data along with related programs and services. The offerings include phylogenetically ordered alignments of ribosomal RNA sequences, derived phylogenetic trees, rRNA secondary structure diagrams and various software packages for handling, analyzing and displaying alignments and trees. The data are available via FTP and electronic mail. Certain analytic services are also provided by the electronic mail server. 3. Silver provides comprehensive, quality-checked and regularly updated data sets of aligned small and large subunit ribosomal RNA sequences for all three domains of life. 4. GRENGENS. The GRENGENS web application provides access to the 2011 version of the GRENGENS 16S rRNA gene sequence alignment for browsing, blasting, probing, and downloading. The data and tools presented by GRENGENS can assist the researcher in choosing phylogenetically specific probes, interpreting microarray results, and aligning annotating novel sequences. References. External links: University of Washington Laboratory Medicine, Molecular Diagnosis Bacterial Sequencing, the Ribosomal Database Project, 1, 2, 3, 4.